We're going to start into our lightning talks now. So first up, I'd like to introduce Kian Hong Kock, who will be presenting on the Asian Immune Diversity Atlas. And I'll let you take it away.
Thank you, Arathi. Hi, everyone. Good afternoon. Thanks for the opportunity to speak today. My name is Kian Hong, and I'm presenting on behalf of the Asian Immune Diversity Network. So if you've gone for a blood test recently or gone for your annual physical and received your results, one of the first things you might Google is the reference range of values for your test of interest. You might have noticed in your Google search results that males and females actually differ in some of these tests. For example, females actually have a lower reference range of values for hemoglobin concentration and also for hematocrit, or the proportion of red blood cells or of all cells. So this is just one example, and we've reviewed this and many other dimensions of human diversity, and we find that these actually have a pervasive influence on biological and biomedical phenotypes. And you can read more about these and some of the directions we're thinking about in terms of single cell and genetic diversity in our roadmap, linked through the QR code over there. For example, if we think more generally about dimensions of human diversity such as ancestry, we find that the United States FDA recommended a lower dose of rosuvastatin, which is a statin used for lowering LDL or bad cholesterol in Asian patients. And for age, telomere lengths actually decrease with increased biological age. So I think all of these are just anecdotes to say that any reference or baseline really needs to take into account different dimensions of human diversity. So many of the investigators that are part of the Asian Immune Diversity Atlas Consortium actually came together wanting to ensure that right from the inception that the Human Cell Atlas was diverse and inclusive of Asian population donors from Asia, which actually collectively constitute around 60% of the global population. So we started with Singapore, Japan, and South Korea, added in India and Thailand, and more recently have countries have member teams from Russia, Sri Lanka, and Pakistan. And collectively, our members aim to construct the world's first single-cell reference atlas of healthy donors from Asian populations. So we'd like to use this atlas to elucidate the landscape of variation, starting first with circulating immune cells, specifically peripheral blood mononuclear cells, look at the effect of human diversity on cellular phenotypes, and use this as a suitable healthy baseline for disease comparisons of Asian populations in the many precision medicine initiatives that are taking flight in Asia. So we've got several of the AIDA investigators represented here today. You might have heard from Partha Majumdar from our AIDA India team. We've got Po Xiang Liu from AIDA Splicing, and also Jay Shin, whom you hear more about in his talk on day 3. So in addition to the diverse Asian populations groups we have in the Asian Immune Diversity Atlas, we've also got a wide range of analyses, from the reference atlas for Asian populations led by Sham, Weng Yang, Jay, to immune repertoire diversity led by Weng Yang, regulatory activity led by Jay and Chung Chen Hao, and splicing by Po Xiang Liu, and also quantitative trait loci analysis by Yuki Okada, Kazuo Ishikaki, and Muin Choi. My aim in this lightning talk is to give you a flavor of the reference atlas to highlight how we're thinking about human diversity in the context of single-cell phenotypes. So we've got the first phase of AIDA, and this is just depicted here in this UMAP, more than 1 million peripheral blood mononuclear cells from more than 500 Asian donors. And we've been able to annotate more than 30 fairly core cell types and elucidate the effects of these covariates that we're interested in on single-cell phenotypes and proportions. So for example, if you look at major immune cell types across sex, I mentioned the sex differences in hematocrit and hemoglobin concentration. We see that in our data set, B cell numbers are higher in females, while NK cell numbers are higher in males. And this is consistent with a lot of the prior work done by immunologists, say using FACS data. But if we try to layer on really the influence or the consequences or the effects of ancestry, we find that we are starting to identify these interaction terms between these two covariates or dimensions of human diversity. So for example, if we look at naive B cell proportions in our ancestries broken down by sex, we still see that in all of the 5 ancestries we have represented in the first data freeze of AIDA, we've got higher numbers or proportions of naive B cells in females compared to males. However, if we look at the individual ancestries, and I've summarized this through this interaction plot over here, we find that the difference appears to be greatest in the Singaporean Indian donors in females versus males. So this we see as an interaction between sex and Indian ancestry on naive B-cell proportions. I'm going to show you another example. So if you can look at NK cells, where I mentioned earlier that males have got higher proportions or numbers of NK cells as compared to females. So if you look at the 5 ancestries that constituted the first data freeze, we don't see much of a difference in terms of across the ancestries, in terms of the CD16 plus NK subtype proportions. But if you look at the breakdown by sex and ancestry, in 4 of the 5 ancestries from the Chinese, Indian, Japanese, and Koreans, we see this higher B-cell proportion, this higher NK cell proportion in males versus females. But in the Malay ancestry in Singapore, so donors of Malay ancestry in Singapore, we find that the Malay ancestry essentially negated the sex difference that we see in terms of NK cell proportions. So that's actually summarized by the interaction plot over here. And just one example of the kinds of a different type of interaction we see between sex and ancestry on NK cell proportions. With the advantage of, or the resolution afforded to us by single-cell transcriptomics, we can look for cell state signatures of human diversity. I'm just going to show you one example over here where amongst our myeloid cells, there's a particular CD16 plus monocyte cell state that's enriched in the older AIDA donors above 55 years old and has a particular gene signature. So we think that it's important to consider these ancestry, age, or sex-specific cell state enrichment, particularly when groups are thinking about disease profiles or single-cell profiles of disease, to avoid trying to confound these covariates of human diversity with disease-specific profiles. So in this very short lightning talk today, I've told you about the Asian Immune Diversity Atlas and the world's first single-cell reference atlas of healthy donors from Asian populations residing in Asia. We're continually expanding our cohort, so in our latest data freeze, we've got more than 1.2 million PBMCs from more than 600 Asian donors, spanning 6 ancestries in India, Japan, Singapore, South Korea, and Thailand. And in the next 2 years, we're looking to expand this to a total of more than 2,000 donors across the 8 member countries that I mentioned at the start of the presentation. I've shown you some examples of the interplay between dimensions of human diversity on single-cell phenotypes, and also how we're thinking about cell state signatures associated with dimensions of human diversity. I'd like to thank many people in the AIDA consortium, especially Sham, Wongyang, and Jay, who initiated the AIDA project, my team in Singapore, and the Genome Institute of Singapore. And it's really been a great privilege and pleasure to work with many talented researchers and investigators in the AIDA consortium. And here's a picture of us at the HCA Asia Bangkok meeting during our inaugural AIDA workshop last year. So thank you for listening, and I'll be happy to take any questions, and also to connect with you if you have any thoughts or would like to know more about the Asian Immune Diversity Atlas. Thank you.
I think we have 2 minutes for questions.
Malta?
Thanks a lot, Kian Hong. Really cool overview. I really liked the motivation at the beginning. You mentioned in the motivation, you said kind of hemoglobin levels are different in males and females, and you kind of showed cell type proportions. Do you expect this all to be cell type proportion shifts that you kind of show, or also have you looked at cell state shifts or state changes?
Yeah, so we've looked at cell state differences in our Asian population groups, and I think there we have to just be a bit more careful because, as I think Sham or some people might mention, there are these batch effects that could act as confounders. So what we are most confident in right now in terms of ancestry-specific population comparisons are, say, internal controls, male versus females, or within the Singapore cohort where we've got 3 ancestries profiled in one country. But that's definitely one of the things we're looking at. Thanks, Malta.
One question back there.
Take Tuli.
Very nice data and great talk. I wanted to kind of like ask what kind of plans do you have for collecting more rich metadata of sort of health and disease and environmental exposures and all these kinds of things? Like I think that as sort of like a comparison in the US, there has been this kind of where it's easier to get, let's say, more uniform and richer metadata in let's say, in different healthcare systems. There's been some interesting work showing that some of the kind of differences that seem ancestry specific are actually kind of like come from socioeconomic drivers of health and disease and different environmental exposure, for example, to pollution, etc. That is not really about that ancestries are different. It's more that different kinds of risk factors are different and affect people in different ancestries in different ways.
Thank you for the question, Tuli. And I think one of the things we're cognizant about is that when we say these are the influences of ancestry, it's really, there's a lot of things tied in together. So there's correlation with diet or with lifestyle. So the first question is, we do collect fairly rich metadata. So we've got a list of questionnaires, of questionnaire fields. And we're also pairing with the precision medicine program in several of these countries. So in Singapore, we're working with the National Precision Medicine Program, SG100K and Helios. So we've got really a wealth of metadata. I think, we do have to take these in mind or regress them out when doing these ancestry-specific analyses. So thank you for that, and that's something we're definitely thinking about.
All right, we are unfortunately out of time for questions. Another round of applause for Kian. And next up, we have Collins Morang'a, who is joining us, I think, virtually. Awesome.
Can start. Thank you very much. My name is Collins Morang'a, and I'm going to talk to you about my work that I did during my PhD on single-cell RNA-Seq, where we showed that we have elevated interferon responses and TNF-α signaling by NFkB in monocytes in children with clinical malaria infections. I'm from the West African Center for Cell Biology of Infectious Pathogens. Basically, malaria still remains a global challenge, whereby more than 3.2 billion people remain at risk of Plasmodium infections. And one thing that we do not very well understand is the complexity of immune cell responses, because sometimes you can find children who have asymptomatic infections or subclinical infections, and they continue with their activities, going to school and all that, yet you find other children who fall sick from the same infection, most likely from the same community, and they end up being hospitalized. And some of the things that our scientists have defined over the years is that sometimes you have resistance to infection or tolerance to infection, but really the cellular level immune responses during clinical malaria continue to be a challenge, and this prevents us from the development of better vaccines that can be able to prevent this disease burden. And so because of this, we sought out to do single cell work so that we can be able to understand what differentiates these different clinical phenotypes and be able to characterize at the cellular level differences in gene expression that determine these clinical phenotypes. To do this, we designed a study which we obtained ethical clearance and did a cross-sectional study where we collected samples from this one region. And the reason for this was that we wanted to minimize the impact of differences in transmission intensity. So we collected samples from one region, and we collected 224 samples, 90 subclinical and 134 clinical samples. And when we evaluated the parasite densities or the relative dominance of some of the variables we collected, these include both socio-economic factors as well as parasite density levels, the age of the participants. We realized that you can see that fever, headache, parasite density were clearly dominating clinical malaria infections compared to subclinical infections. And when you do combination of these variables, you realize that when you combine parasite density, fever, and headache, they generally tend to have a higher dominance in terms of clinical malaria infection. And because of this, we also analyzed, so what is parasite density, the effects of parasite density across the different ages of the participants that we collected. And you can see that generally for the subclinical infection, which is blue, there was no correlation. But the clinical, there was significant correlation, whereby children under 20 years, they tend to have higher parasite density levels, which tends to decline across time. And furthermore, if we break down this age group, these ages into different age groups, you realize that majority of majority of the individual above 16 years old, there's no significant difference between their parasite densities, as well as children below 5. But between 6 and 15, there is significant difference in terms of the parasite density, whether you are clinical or subclinical, and the clinical tend to have higher parasite densities compared to this. So we decided to pick on individuals within this category for our single-cell sequencing, and we took their PBMCs. These are 5 subclinical individuals and 6 clinical malaria individuals, whereby we try to normalize in terms of their clinical phenotypes. So we did single-cell RNA-seq using SQL, and we analyzed this data set in R, and as well as we did some level of cell-to-cell interaction analysis. In terms of analysis, we did quality control, clustering, cell annotation, and differential expression analysis and cell-to-cell interactions. We identified 10 different clusters in about 18,000 cells, and we annotated these cell types using common cell markers, cell annotation markers. But to further annotate these cell types, we did a reference-guided analysis whereby we used a CiteSeq reference dataset from Howe et al. And with this, we were able to get a higher granularity in terms of the different cell types that we had in our PBMCs. And when we did fractional abundances, we realized that only B cells had significantly higher cells in subclinical compared to the clinical, while the monocytes were significantly higher in the clinical individuals compared to the subclinical. And down into their minor cell types, you can see that the different B cells and as well as the monocytes, the CD16 monocytes and 14 monocytes were significantly higher in the clinical malaria individuals. And when we checked on the differentially expressed genes, we realized that what we are showing is that the up are in red and the down regulated are in yellow. You can see that we have quite a number of different genes that are upregulated in the clinical malaria individuals. And when we do pathway analysis, we see that we have several pathways that are upregulated in clinical malaria individuals or enriched. And we have several of them that are involved in immune response, such as complement, inflammatory response, P53 pathway. And the most interesting ones for us were the interferon, alpha and gamma, and NFkB signaling pathways that were significant. And when we analyze the module scores for these interferon-stimulated genes, you can see that clearly the monocytes have significantly higher interferon-stimulated genes compared to the subclinical, as well as the B cells, the CD4 T cells, and the rest, as well as the monocytes in the clinical malaria infections. All the different NFkB target genes were tended to be significantly higher compared to the subclinical individuals. Further, we did cell-to-cell interaction networks, and you can see that visually comparing the clinical malaria and subclinical, we have more interactions in the clinical malaria samples compared to the subclinical. And this, you can see that if you evaluate the total number of interactions between the different cell types, these clinical malaria individuals tend to have higher cell-to-cell interaction between the different cell types, especially the monocytes towards the CD4 and CD8 T cells. And when we looked at what exactly was determining these cell-to-cell interactions, we realized that majority were MHC signaling pathways. These are all the different pathways that were involved. And you can see that the MHC class 1 and class 2 were majorly involved in the clinical malaria compared to the subclinical individuals. And I would like to summarize that what we've shown is that we have identified the different cell types that are significantly different between clinical and subclinical infections, and there was significant higher fractional abundances in monocytes of clinical malaria. And several cytokine genes were upregulated. These are interferon responses, TNF-α signaling, and we've also shown that monocytes interact with different cell types in driving cell-to-cell interactions. And this is one of the initial malaria immune single-cell data sets for defining clinical and subclinical infections. And we hope that in future, we are going to explore these mechanisms in severe malaria individuals, as well as conduct longitudinal studies in healthy people, subclinical and clinical malaria, so that we can further develop a malaria disease atlas. I would like to acknowledge my supervisors, as well as acknowledge Alex Shalek, Lab, and Vincent and Riley, who assisted in conducting these experiments. Thank you very much.
All right, we have 2 minutes for questions.
Hi, Hari Nakshatri from Indiana University. Have you done the Duffy genotyping of those with the subclinical versus clinical malaria, whether some of the interferon, some of the immune responses could be, differences could be because of the Duffy phenotype?
No, we haven't done the Duffy genotyping, but we could consider doing that to be able to understand them. Yeah, thank you.
Hi, thank you so much for this talk. I had a question about you focused here on children, but you said that also in adults you observe these like clinical and subclinical phenotypes. So do you think that you would find the same cell types implicated in that type of cohort, or do you think that because there were these differences in like parasite density, then maybe another cell type or another cell phenotype would be mediating differences between clinical and subclinical in adults?
Yeah, so normally what tends to happen with malaria infection is because of repeated exposure. So children who are born in endemic areas, they tend to develop immune kind of responses or tolerance or resistance to infection as they age. So a person living in a high endemic area, by the time they are like 5 years, they've probably been exposed to more than 10 or 15 different malaria episodes, and they've developed immune response. So it means the more you stay in a malaria endemic area, the more you develop kind of different immune responses. And the hypothesis on tolerance to infection is that we still don't know why some adults still come to the hospital with the infection. And there's a possibility that the cell types, as well as the immune responses that they will be exhibiting, might be probably slightly different from what we are seeing in children. So yeah, so it's possible that they might show different cell types as well as different cell gene expression pathways that are different from those of children.
Awesome. Thank you so much, Collins. One more round of applause. Last but not least, we have Daniela Senra, who's going to give the last lightning talk.
Hello, everyone. Thank you for the introduction. I am a PhD student from Argentina, and today I'm going to share with you some research I've been doing as part of my PhD, which is a computational methodology to identify, to quantify pluripotency and biological processes in general. So one of the most common approaches to identify stem cells or any cell type is to use markers. And this approach could be limited due to the sparse nature of this type of data. So here we introduce a methodology to quantify the activity of a protein-protein interaction network that is associated with any biological process from single-cell RNA sequencing data. And in the particular case of cell differentiation, we can identify highly pluripotent cells and use those cells to then perform trajectory inference. And they will be the root, the starting point of when performing trajectory inference. And this tool was named Origins and was implemented as an open source R package. We use two prior knowledges from Pathway Commons. We downloaded all the protein-protein interactions for humans. And on the other side, we used QuickGo from where we downloaded the genes associated with the biological process we are interested in quantifying. In this case, cell differentiation. And here you can see the protein-protein interaction network resulting from merging those two data sets. So once we have built our network, we wanted to find a way to quantify how active that network is. And for that, we proposed a model where genes are the nodes of an undirected graph, and these genes are connected by weighted edges. And these weights were calculated as the multiplication of the expression levels of the genes involved in that interaction. So we added up all the edges, and we obtained a score that represents the activeness of that network. The higher the score, the more active that network would be for that cell. You can see the same in math language. So we also included other pre-built protein-protein interaction networks associated with other biological processes. Here are some of them listed. And we also included a function to build a network associated with any gene set that the user might be interested in. So once they build the protein-protein interaction network, the user can calculate the activity. And we benchmark our tool with 4 healthy human data sets. And we compared 4 methodologies, including ours. And we assessed different performance parameters, such as computing time, RAM usage, input flexibility, among others. You can see more detail about this benchmarking process in our publications. And to summarize, we created a computational methodology to calculate the activity of a protein-protein interaction network. The inputs are an expression matrix and the biological network we want to quantify the activeness. And the output is a vector of activities. So each element of that vector represents the activity for each single cell. The higher the activity, the more active that network will be. So here is an application to a healthy human breast data set. You can see cells in the PCA space. Cells are colored according to differentiation activity. Here were the cells with the highest activity. So we use those cells as the root to perform trajectory inference, and we did that here, and we obtained its spectral results, branching differentiation trajectory, starting from the basal cells to luminal differentiated cells. And here you can see a correlation matrix of all the biological processes we quantified the activity. So here is an application to a breast cancer data set. In particular, this is a triple-negative breast cancer sample. And you can see cells in the UMAP space. Cells are colored according to different biological processes, activities. We quantify, for example, stem cell differentiation. We found out a small group of tumor cells that have high activity levels of stem cell differentiation. We also found out a subset of cells that have a high cell cycling activity, which is expected right here. And for example, for adaptive immune response, we found out that tumor amplifying macrophages have the highest activity score. So, if you want to know more, here are the publications. The data we used is listed here, and we made available this code as an open source R package. You can access it from my GitHub profile, and here is the QR code, so you can access to it. And well, I want to thank the organization, the Open Bioinformatics Foundation, and CONICET for their support, and my advisors, Luis Diambra and Nara Isoni. So thank you very much.
All right, we've got 2 minutes for questions.
Thanks a lot for your talk. I was wondering, how sensitive is your method to false positives and false negatives in the gene ontology associations to genes, where you kind of generate your specific networks? Have you tested this?
Yes. So the biggest problem would be when the network is small, because data is sparse. So you have lots of zeros, and this multiplication would be a zero. So for small networks, that would be the problem. And in fact, that we benchmark that, yes.
Any other questions? All right, one more round of applause for Daniela.
Thank you.
Thank you so much to all three speakers, and I believe I'm going to hand it off to John.
Hi, everyone. I get the last word. So we've had an amazing day today, really amazing science talks. We had some really thought-provoking discussions about the challenges and the opportunities for building atlases. And there are still 3 ways that you can engage with HCA yet today. Number 1, right after this, we are all going to gather right outside these doors, and we are going to take a group photo. So please don't go anywhere. Well, actually, no, do go somewhere. Go outside the doors, but then don't go anywhere once you get there. And the staff will direct you, and we'll make sure that we can get this done efficiently. Number 2, the reception starts right after that. And number 3, in parallel with the reception, there is the poster session, which will actually be on the 2nd floor. I have been told the 2nd floor is a public space, so you will not be allowed to take alcoholic beverages to the 2nd floor. So just watch that. But the reception, photo first, and then the reception and the poster session to follow. Thank you all very much for attending. It's been a great day, and we look forward to seeing you tomorrow.